I'm Shaila Jain, Assistant Professor at B.Pharmacy College, Saga. Today, I'm really privileged to present another interesting and learning episode of B.Pharma entitled as Neuromuscular Blockers on behalf of Mr. Sandeep Srivastav, Assistant Professor at B.Pharmacy College, Saga. So friends, we'll start with a general introduction of the topic followed by structure-activity relationship, mechanism of action, neuromuscular blocking agents, classification and depolarizing blocking agents. Module 1. Introduction to neuromuscular blockers. Drugs that relax skeletal muscles are called skeletal muscle relaxants. These agents are classified according to the use and mechanism of action. These agents produce muscle paralysis required for surgical procedures and hyperactivity. Neuromuscular blocking agents block the transmission of acetylcholine at the motor end plate, so considered as nicotinic antagonists. The therapeutic use of these compounds is primarily as adjuvants in surgical anesthesia to obtain relaxation of skeletal muscle. They are also used in various orthopedic procedures such as alignment of fracture and correction of dislocation. The neuromuscular junction consists of the axon imposing onto a specialized area of the muscle known as the muscle end plate. The nerve terminal is separated from the end plate by a gap of 200 angstrom. Now, uses of neuromuscular blocking drugs. First one is endotracheal intubation. Second, reduce muscle contractility and depth of anesthesia required for surgery. Third, in the intensive care unit to prevent high airway pressures, decreases oxygen consumption and abolish muscle rigidity in patients on mechanical ventilation. Last is reduces the chances of bone fractures during electroconvulsive therapy. So friends, here we have ended our first module and proceed to our next module. Module 2. Structure and structure-activity relationship of NMBDs. All the NMBDs are available are quaternary ammonium compounds. The type of alkyl carbon, that is methyl or ethyl, indicates the charge distribution and binding characteristics of onium compounds. Number 2. Non-depolarizing drugs are generally bulky and more rigid than depolarizing drugs. Number 3. As the steric hindrance to receptor increases, the potency decreases. Number 4. The levorotatory tubocharrin is comparatively less potent than the dextrorotatory tubocharrin. And the last point is the depolarizing agents, for example, decamethonium, have a more flexible structure that enables bond rotation. The distance between quaternary groups in the flexible depolarizing agents can vary up to the limit of the maximal bond distance, that is 1.45 nanometers for decamethonium. They are structurally related to acetylcholine. As with acetylcholine, the positive nitrogen atoms of NMBDs are attracted to alpha subunits of the postsynaptic nicotinic receptor. Many NMBDs, for example succinylcholine, pancuronium and atracurium, contain 2 quaternary ammonium cations. And the last one is these bis quaternary amines are more potent than monoquaternary amines, for example ochironium, tubocharrin and vecuronium, which have only one permanent quaternary cation and one tertiary amine. At physiological pH and especially in acidotic conditions, the tertiary amine can become protonated and therefore positively charged, increasing the potency of monoquaternary NMBDs. This factor has clinical significance. The 2 quaternary ammonium groups are separated by a bridging structure that is lipophilic and varies in size. The bridging structure varies with different series of NMBDs and is major determinant of the potency. Module 3. Mechanism of action of neuromuscular blocking agents, drugs. Normal physiology at neuromuscular junction. Friends, here we will discuss the figure of acetylcholine channel. As the acetylcholine become attached, leads to conformational change, thereby causing the opening of channel, allowing sodium ions to enter the muscle fiber and excite contraction. Acetylcholine activates mainly 2 types of receptors called muscarinic and nicotinic. Muscarinic receptors are found on all effector cells that are stimulated by the postganglionic cholinergic neurons of either the parasympathetic nervous system or the sympathetic nervous system. Nicotinic receptors are found in the autonomic ganglia at the synapses between the preganglionic and postganglionic neurons of both the synaptic and parasympathetic systems and the neuromuscular junctions in skeletal muscle. Skeletal muscles are innervated by somatic motor nerves which originates in the spinal cord, terminate at muscle cells and release acetylcholine as the neurotransmitter. Upon arrival of an action potential, acetylcholine is released from synaptic vesicles by exocytosis, crosses the synapses and interacts with skeletal muscle nicotinic cholinergic receptors to depolarize the postsynaptic membrane. When the membrane gains acetylcholine threshold, a muscle action potential is generated and propagates along the fiber to initiate excitation-contraction coupling. Neuromuscular blocking agents interfere with neurotransmission by either of the mechanisms. Number 1. Competitively antagonizing the actions of acetylcholine at nicotinic receptors which occur with the non-depolarizing agents, and the second one, that is by occupying and activating the nicotinic receptor for a prolonged period of time leading to blockade which occurs with the depolarizing agents. Drugs can block neuromuscular transmission in three main ways. The first one is by inhibiting acetylcholine synthesis, for example, hemicholine and triethylcholine. The second one is by inhibiting acetylcholine release, for example, streptomycin, neomycin and two potent toxins, namely botulinum toxins and beta-bungarotoxins. The third mechanism is by interfering with the postsynaptic action of acetylcholine. This category can be further divided into two main classes. The first one is non-depolarizing blocking agents and the second one depolarizing blocking agents. So friends, in this figure, you can see the neuromuscular transmission and sites of drug action. The action potential is conducted down the motor nerve axon and can be blocked by sodium ion channel blockers such as local anesthetics or tetrodotoxin. So as you can see in this figure, when the action potential reaches the nerve terminal, calcium ion mediated vesicular acetylcholine release occurs by exocytosis, a process blocked by botulinum toxin or hemicholine. Then after release, acetylcholine diffuses across the synaptic cleft and binds to nicotinic receptors in the motor end plate. The binding can be blocked competitively by non-depolarizing neuromuscular blockers such as curare or non-competitively by depolarizing neuromuscular blockers such as succinylcholine. Acetylcholine is very rapidly hydrolyzed by acetylcholine esterase which can be inhibited by choline esterase inhibitors such as neostigmine. Then acetylcholine activated nicotinic receptors in the motor end plate generate a muscle action potential which leads to calcium ion release mediated by ryanodine receptors, initiating excitation contraction coupling and muscle contraction. This calcium ion release can be blocked by anti-spasticity drug, that is dantrolene. So friends, before proceeding to our next module, I would like to suggest you to kindly visit our website, that is www.cec.nic.in. Here you will find many lectures, e-content, FAQs, LORs and many more. I hope this website will be a catalyst of learning for you. Module 4. Classification. NMBDs are classified as depolarizing and non-depolarizing drugs according to their action at the post-junctional nicotinic receptor, non-depolarizing or competitive blocking drugs. Non-depolarizing blocking drugs compete with acetylcholine on the postsynaptic nicotinic receptor. Binding to one or both alpha subunits prevents access by acetylcholine to depolarize the receptor, so end plate potential becomes very small to initiate the propagated action potential. This results in paralysis of neuromuscular transmission. Non-depolarizing blocking drugs do not produce conformational changes in the receptor. Now, as you can see, NMBDs can be classified into three main classes. The first one is long-acting, for example, D-tubercularine, pancuronium and doxycurium. So, firstly we will see about tubercularine. Botanical source of dextrorotatory tubercularine is Condodendron tomentosum, an alkaloid. Tubercularine is the lead compound, contains isoquinoline, phenol and aromatic ring. Tubercularine contains quaternary nitrogen in 2-isoquinoline ring, which is essential for neuromuscular blocking action. Tubercularine is a non-depolarizing blocking agent used for its paralyzing action on voluntary muscle, the site of action being the neuromuscular junction. Its action is inhibited or reversed by administration of acetylcholine esterase inhibitors such as neostigmine or by androphonium. Tubercularine has a long onset of action and a prolonged duration of effect. The main side effect of tubercularine includes histamine release and hypertension with compensatory acetylcholine tachycardia. The next category is intermediate acting, for example, atracurium, vecuronium. Now we will discuss about atracurium. Atracurium is a non-depolarizing neuromuscular blocking agent. It is approximately 2.5 times more potent than dextrorotatory tubercularine. Its duration of action is about 0.33 hours, is much shorter than that of dextrorotatory tubercularine. Pharmacologically inactive metabolite is lodanosine. Atracurium bisylate break down by a Hofmann elimination reaction. Acetylcholine esterase inhibitors antagonize paralysis by atracurium. So friends, here we have reached the third category, that is short acting. For example, galamine triethiodide. Synthesis. Pyrogalol is condensed with 2-chloro-triethylamine and the resultant triamine is quaternarized with ethyl iodide in boiling acetone. Friends, here we have reached our last module, module 5, depolarizing blocking drugs. Depolarizing neuromuscular blocker such as succinylcholine produce prolonged depolarization of the end plate, result in desensitization of nicotinic acetylcholine receptor, inactivation of voltage-gated sodium channels at the neuromuscular junction, increases in potassium permeability in the surrounding membrane. Drugs in the category of depolarizing blocking agents depolarize the membrane of the muscle and plate. They depolarize by opening sodium ion channels. Depolarization shows similar effect like acetylcholine on ganglia and neuromuscular junctions. That is, it is so-called nicotinic effect. This phenomenon is known as acetylcholinephile excess or desensitization. Depolarizing agents produce dual mechanism and blockade occurs in two main phases. The first is phase 1 block. This phase is a consequence of prolonged depolarization, rendering the membrane unresponsive to further stimuli. It is characterized by initial muscle fasciculations followed by a flaccid paralysis that is not reversed but intensified by administration of acetylcholine esterase inhibitors. It is rapid in onset, results from persistent depolarization of muscle and plate. The next is phase 2 block. This phase progresses to a state in which the block appears similar to that produced by non-depolarizing agents. That is, it becomes responsive to high concentrations of acetylcholine and can be reversed by acetylcholine esterase inhibitors. It is slow in onset and results from desensitization of the receptor to acetylcholine. The first is succinylcholine. Succinylcholine chloride has a very short duration of action. It shows quick recovery because of its rapid hydrolysis after injection. Its action with that of dextrorotatory tubercularine is not antagonized by acetylcholine esterase enzymes. Succinylcholine act normally for about 3 minutes and hydrolyzed by plasma cholinesterase. Metabolism of succinylcholine. Decamethonium. Decamethonium has a short duration of action. It is partial agonist of nicotinic receptor. In the process of binding, decamethonium depolarizes the motor end plate, but since the decamethonium itself is not degraded, the membrane remains depolarized and unresponsive to normal acetylcholine release. Decamethonium does not produce unconsciousness and anesthesia. So friends, in this table, you can see antagonism of neuromuscular blocking agents. Here, antagonizing agents are described along with the dose and mechanism of action. For example, the first one is androphonium. In dose of 10 mg, mainly works by inhibiting acetylcholinesterase enzyme. Androphonium again in dose of 0.05 to 0.1 ml per kg works by inhibiting acetylcholinesterase with anticholinergic action. Third is neostigmine. In dose of 0.5 to 1 mg mainly acts by reversible inhibition of acetylcholinesterase enzyme. And the last one you can see pyridostigmine. Pyridostigmine in dose of 10 mg mainly works by inhibiting reversible acetylcholinesterase enzyme. So friends, I hope this table is clear to you. So now in this next table, you can see significant drug interaction with neuromuscular blocking agents. Here, the class or the category of the drug is mentioned along with the clinical effect. For example, antiarrhythmic agents, for example, quinidine, procainamide, lidocaine. They mainly enhance the NMB activity. Antibiotics, for example, aminoglycosides, tetracyclines, polymyxins, clindamycin and piperacillin. Their mainly clinical effect is respiratory depression and excessive blockade. Third one is anti-epileptic, for example, carbamazepine, fosphenytoin and phenytoin. Its clinical effect is rapid recovery time after NMB administration. Fourth is calcium channel blockers, for example, verapamil and nicardipine. It mainly enhances the NMB activity. Fifth category is inhalational anesthetics, mainly enhance the NMB activity. Sixth category is oral contraceptives. It mainly prolong the NMB activity. And the last one, as you can see over here, tricyclic antidepressants. The main clinical effect is risk of ventricular arrhythmia. With all these information, here we come to end of today's lecture. Do keep in mind what we discussed today.